So as Paul mentioned, today I'll be talking about diagnostic approaches to detecting COVID-19 with CRISPR. For today's talk, I'd like to provide an overview of CRISPR-based diagnostics and discuss how Mammoth has reconfigured our CRISPR-based detector platform to develop a COVID-19 diagnostics that we call COVID detector. So when we hear about CRISPR, many of us may think of the Cas9 protein and gene editing applications. And although Cas9 has paved the way for genome editing, over the last several years, there has been an explosion of new CRISPR systems that have been discovered and characterized. And many of these novel CRISPR proteins have unique properties that enable an expanding toolbox for CRISPR-based applications. This timeline gives you a sense of where the CRISPR field has started with respect to applications and the work on Cas9, and it's really been in the last 5 years that we've seen a number of new proteins that have come onto the scene with new capabilities that we're now just beginning to understand how to use in these different applications. The programmable nature of these RNA-guided CRISPR nucleases makes it a powerful technology that can be readily modified to target any sequence of interest. And it's this underlying principle of CRISPR, coupled with its natural or engineered features of specific nucleases, that have expanded our CRISPR toolbox beyond genome editing. And these applications can include diagnostics as well as genome modulation. But for the purposes of today's session, we'll be focusing on how we use these CRISPR enzymes and technology for enabling new methods for molecular diagnostics. In this way, we can repurpose CRISPR as a search engine that can find and detect any nucleic acid target. And the way this works is that upon finding that matching sequence, the CRISPR complex is guided by the sequence in the guide RNA to target the specific DNA or RNA molecule. When that protein finds the matching sequence, it turns into a non-specific nuclease, which is able to cleave reporter molecules to release a signal that provides a real-time diagnostic result. And in this example, this reporter molecule contains a fluorophore that's linked to a quencher by a single-stranded DNA or RNA molecule. And so when this molecule is intact, the reaction remains dark, and when it's cleaved by the CRISPR protein, it produces a fluorescent signal. And the distinct families of CRISPR proteins that have this diagnostic capability include the Cas12, Cas13, and Cas14 enzymes that naturally target DNA or RNA sequence. And this enables a wide range of flexibility when we're designing diagnostic assays. So in the next slide, let's take a journey inside a test tube to visualize the chemistry. So in this video, you can see that we first pair a Cas protein with a guide RNA that is programmed to match the exact sequence we want to detect. These molecules form a complex that searches through billions of letters to find, in this case, cut the matching DNA or RNA sequence. But the unique piece of these CRISPR proteins is that they don't stop there. You can see this nuclease is turning on its nonspecific activity that is chopping up nearby pieces of DNA or RNA. In this case, it's the reporter molecule that creates a color change to let us know that we've found our target sequence. Our CRISPR-based diagnostic platform called Detector is a sample-to-answer workflow that involves first a crude sample preparation step, followed by a rapid and multiplexable preamplification that achieves single molecule sensitivity, and then followed by the CRISPR detection module that sorts through non-target sequences with single nucleotide specificity. And so as an integrated system, Detector is simple and it's faster and has similar performance compared to the gold standard PCR methods. And together this is really enabling new ways that we can multiplex, detect different genomic targets, single nucleotide polymorphisms, and a lot of different flexible ways to approach detection using the CRISPR enzyme itself to recognize a nucleic acid target. The flexibility and simplicity of the Detector platform allows it to be used across a continuum of testing solutions. And so on one end, a single-use disposable molecular test has a potential to place the accuracy of a molecular lab in the hands of consumers, which would enable decentralized testing to detect diseases earlier and in vulnerable environments. And then diagnostic testing in central laboratories are also needed to process large sample volumes with high performance, and workflow improvements would enable even greater testing throughput. And what's really exciting about the CRISPR technology is its capability and potential to advance technologies across this testing spectrum. So now I'll transition into some of the work that we've been focused on leveraging this detector platform for COVID-19 diagnostics. So many of us are familiar here with the virus starting to make its way into the US in early February and March. And what you can see here are, a very small subset of articles that were coming out describing the massive problems related to coronavirus testing that caused major bottlenecks in deploying an effective pandemic response, ranging from a dearth of coronavirus tests to poor reagent quality, supply chain issues, logistic difficulties, and the list goes on. And a lot of these early missteps really, affected our efforts to stay ahead of the viruses, and many of those challenges really outstripped our ability to really deliver on an effective strategy. Unfortunately, we're now, 6 months into the pandemic, and it's interesting watching the same articles or similar articles discuss, again, the challenges that we continue to face. Hundreds of thousands of lives have been lost. We still lack a coordinated public health strategy. Now we're also facing a situation with demand for coronavirus testing far exceeding supply. We still will face challenges that hold back a fully scaled testing strategy that returns results within hours instead of days. We're seeing even the major laboratories in the United States face turnaround times of multiple days, up to even a week in some cases. And I think this, coupled with mixed messaging around public health guidance, shifting behaviors, we face a situation where the virus continues to rage as we move into the fall. A lot of this, there's going to be multiple responsibilities, and there's not going to be just a single magic bullet. And what I'd like to focus on today is the efforts of the diagnostic approaches towards combating the pandemic. So this is a figure taken from the Johns Hopkins Coronavirus Response Center that illustrates daily testing trends across the US. This is a screenshot taken yesterday, and although the number of tests have ramped up significantly compared to the spring, we still fall short of reaching the millions of tests per day target that is estimated to enable suppression level testing that is necessary to stay ahead of the virus. One potential caveat of this data is that it may not include distribution of the new instrument-free antigen tests that have recently been launched, and so that might not be accounted for in this particular data set. But one thing I want to point out is that actually for the first time since June, the percent of positive tests was below 5%. Earlier this week, I think as of yesterday, it went up to 5%. And so this is an important milestone, which is likely due to a combination of public health efforts and the increasing proportion of high-risk patients that are already being infected. One important note is that the WHO has said that in countries that have conducted extensive testing for COVID-19, this number should remain at 5% or lower for at least 14 days. So we're not quite at that level of testing, it seems, but the data collected through the end of this month into October will help us better understand the trajectory of the pandemic as we enter the fall. So there are several technologies that are being used currently to detect the virus. So they can be kind of summarized in these 3 different categories. The first is nucleic acid testing. And this is a method to directly detect the RNA of the virus. What you can see in the figure on the right are some of the relative detection loads and probability of detection using each of these methods. At the moment, the gold standard PCR-based tests do provide the earliest window for accurate diagnosis, and you can see that the kind of range of time at which this test is most effective really is in the first couple weeks of infection, and potentially in asymptomatic patients before symptoms arise. The other category of detecting the virus include antigen-based testing. So again, this is another direct approach that tests for specific viral proteins of the virus. And these are typically lower sensitivity, but they are very rapid and equipment-free. Most of these are useful for surveillance testing within 1 week of symptom onset. So they are quite effective, a week after symptoms are presented. And finally, the last category is antibody testing. So this can be thought of as an indirect testing approach that looks at the host immune response to the viral infection. And serology testing, as you can see in this graph, is typically later in the progression of disease, and this is usually most useful for surveillance of the exposed. So all these tools are important for us to be able to understand progression of disease, the outbreaks that are occurring, but wanted to recognize that there really is a combination of approaches that are necessary, and the work that I'll be talking about using CRISPR really falls into the first category of actually detecting the nucleic acid of the virus itself. So shifting into some of the work at Mammoth, this timeline is a snapshot of some of the key events that occurred during the early days of the SARS-CoV-2 outbreak in relation to the development of our detector assay. One interesting piece of history is that we started looking at this virus before there was an official name for the pathogen or the disease, and it really was early on that we figured that it was important, and we had the potential and opportunity to use our detector system to go after SARS-CoV-2. And it took us about 10 days from designing the detector assay to testing the reagents on one of the first COVID-19 patient samples in the US that was provided by our collaborator, Dr. Charles Chu from UCSF. And you can see here that it was really, after the first confirmed case in the San Francisco Bay Area, and thanks to the collaboration with Charles, we were able to confirm our early assay on real patient samples, which is very important in the course of test development to be able to look at real clinical samples. Last month, Mammoth received EUA approval for our SARS-CoV-2 detector reagent kit that is authorized for emergency use in high complexity CLIA labs. And a lot of this work was based on the early development that was done in the project that I'll be talking about. And in the upcoming slides, I'll talk about how we actually reconfigured our detector platform to target the novel coronavirus. So as I showed in the previous slide, within days of the first confirmed COVID-19 case in the US, we initiated the collaboration with Charles's lab and other colleagues at UCSF to develop a CRISPR-Cas12 based assay for the detection of SARS-CoV-2. So at the time, we used RT-LAMP as our pre-amplification method and designed Cas12 guide RNAs to be compatible with the established WHO amplicons as well as the CDC amplicons that were targeting the E gene and N gene respectively. So these were the tests that were available early in the year, and we wanted to enable that a solution that we were able to develop could be compatible with the existing amplicons, as well as working with off-the-shelf components such as LAMP reagents, as well as Cas12a that could be purchased from commercial vendors as part of the development of this protocol. So this image is a schematic of the full workflow. On the left, we start with what was the gold standard at the time, nasopharyngeal swab. Thankfully, we have a number of new sample types that have been used for sample collection that is less invasive, so including nasal, lower nasal, and saliva. But in this particular work, starting from the MP swab, we transitioned that into a manual extraction method to release the viral RNA. This extract is then transferred to the isothermal amplification consisting of the RT-LAMP. At the time of this protocol, we had been looking at both the E and N gene as well as RNase P as the sample control. The CRISPR protein, again, has been programmed to detect these particular sequences that release the target, that release the probe cleavage event. And what this is also demonstrating is the ability to have the reaction compatible with various readout formats. So on the top is a lateral flow-based point of need format that can be used to actually visualize the reaction. And on the bottom is an example of a Qpcr machine that can be used, so standard lab equipment to read out the fluorescence signal. And so these are different ways that we've been able to modify the detector readout that is useful for various environments. And the work that has been EUA approved is based on the standard lab equipment as kind of described on the bottom here. So to dive into a little bit about the flexibility of the CRISPR reaction, due to the high specificity of CRISPR enzymes, we're able to design guide RNAs that distinguish SARS-CoV-2 from other closely related strains, including SARS coronavirus and a bat SARS-like coronavirus strain. And what you can see here is that we're able to design guide RNAs to distinguish these closely related strains, but in the same vein, we can also design guide RNAs that are capable of what we call broad spectrum targeting of these related strains. And this is another example of a pan-coronavirus detection strategy if the goal is to detect the presence of any coronavirus. So there is flexibility in a pan-coronavirus detection as well as a strain-specific detection using modified guide RNAs. Here we're showing the evaluation of, again, specificity of the SARS-CoV-2 specific guide RNAs and testing them against a panel of other samples that have been infected with other human coronavirus strains. And as expected, we are able to detect only the controls that contain the N gene or the E gene that are specific to the guide RNA, and these other human coronaviruses that are not SARS-CoV-2, we're only detecting the RNase P or sample control from the human RNA. And so this is a nice example of the lack of cross-reactivity to other human coronaviruses with this workflow. We've also adapted the COVID detector assay to commercially available lateral flow strips, as I described in the overview slide, and this is done by simply modifying the reporter molecules to be compatible with the lateral flow strips. And so instead of a fluorophore linked to a quencher, here we have a fluorophore or some other molecule that's linked to biotin. And when the molecule is intact, it remains trapped at the capture line. And upon recognition of the matching sequence, in this case SARS-CoV-2, the Cas12 protein cleaves that reporter molecule, which is now able to migrate to the test line. And that's how we're able to achieve separation of the positive versus the negative sample as seen on this lateral flow strip. We also show that we're able to get a robust visual signal within 5 to 10 minutes of the reaction, as you can see with this time course on the right. This image is just to give you an overview of data that was collected on lateral flow strips on 10 COVID positive and 12 COVID negative patient samples, and what you can see are very distinct bands indicating the presence of the virus or the absence, including the RNase P sample control. And we eventually tested our assay on 83 patient samples, which is the largest sample size, using CRISPR-based diagnostics for SARS-CoV-2 detection at the time of publication. And this COVID detector assay demonstrated 95% positive predictive agreement and 100% negative predictive agreement in comparison to the CDC Qpcr test. It's also been able to provide a faster turnaround time, less than an hour, and has been shown to be compatible with a visual lateral flow readout. And so since the publication of these results, where is Mammoth going from here? So I just want to briefly touch on some of the initiatives that we're working on, again, to target the spectrum of testing needs. And so for decentralized environments, Mammoth is pursuing a handheld disposable diagnostic test that has the potential to transform the paradigm of infectious disease testing. And in May, we announced a partnership with GSK to develop a CRISPR-powered device that places the accuracy of a molecular lab into the hands of consumers. And we're very excited about this opportunity that it will able to circumvent traditional doctor's office diagnosis and enable intervention much sooner, and also prevent further spread of the infection. And overall, this fully decentralized testing model, we believe, will dramatically improve the response to an infection at both the individual and community level. Now, for centralized environments such as laboratories that are limited by capacity, we are also developing a turnkey workstation that will use our CRISPR-based SARS-CoV-2 molecular assay that's based on automated liquid handling equipment from major vendors. And this high throughput system will be able to perform greater than 4,500 tests over 24 hours with minimal user interaction. In July, we were one of the first 7 companies selected to receive a phase 2 contract from the NIH RADx program, which will support the manufacturing and scaling up of this high throughput detector system. And by implementing the sample to answer workflow, CLIA labs will significantly improve their capability to test large groups of symptomatic and asymptomatic people, which we believe will increase SARS-CoV-2 testing capacity, provide the necessary safety and security to address critical aspects of the economy. And finally, I would just like to wrap up my talk with the humbling reminder that there really is a long fight ahead to stay ahead of the virus. And despite the ongoing challenges, it really has been extraordinarily inspiring to be part of a scientific community and movement that's working tirelessly to help curb the pandemic. And with that, I wanted to acknowledge the entire Mammoth Biosciences team for their tireless work over the last several months, and also acknowledge the folks that have been driving the work that I talked about today: James Broughton, Claire Fashing, Xin Miao, Jasneet Singh, and our collaborators at UCSF, Professor Charles Chu, Wayne Dung, and Denise Servalita. With that, thank you for your time, and happy to take any questions.
Okay, please send your questions in through the chat option on the viewer. I had a question related to one of the topics that came up earlier today with the worldwide extent of the problem and the need to address testing across the world. Where do you see this technology fitting into that part of the puzzle?
Yeah, I mean, the goal here is being able to deploy this platform widely, regardless of, the country or location. And so that's why we're taking this dual-pronged approach. There's also, you can think about it as multiple prongs as well, but going both on the decentralized end, where you are able to deploy this kind of accurate testing in vulnerable environments, and then on the other end, where you have the capacity for centralized laboratory to be able to improve those workflows, have a more efficient system that, for instance, uses less tips, has a faster turnaround time. I mean, all these improvements are needed to help keep up. So while we right now, with the RADx program is focused on the United States, really the goal is to develop the solution that can be implemented worldwide. And I think there's going to be a substantial need for that in a lot of these different form factors that I talked about today.
A question we have is, how far off is the single-use device that you are collaborating with GSK on?
Yeah, so I can't talk about specifics, but we are very far into our development program with GSK. And so we hope that, by the end of the year, early next year, there's going to be some exciting announcements around, where that is with respect to the EUA process as well.
A related question you sort of answered. Do you have a projected timeline for this technology to get into the hands of the consumer, both the home testing and then the hospital-based testing?
Yeah, so maybe I'll start with the high throughput testing solution. So We are also, working very quickly to scale up, manufacturing and validating that assay onto these high throughput automation. The goal being that, Mammoth will have the reagent kit that can be deployed on existing laboratory automation that perhaps already exist in clinical labs or the hospitals. So the goal of this is to be able to deploy it, before the end of the year, and then ramp up capacity very, very quickly to address the needs in those environments. And then, as I said, on the handheld disposable, that is a slightly longer timeline given the needs to evaluate how this device will be used with, for instance, unskilled consumers who, have never run a molecular test. It'll be designed in a way such that the user will be able to use it very simply. It'll involve, collection of a nasal swab that inputs into the device itself. So as you can imagine, the more simplistic the user faces, the more simplicity that the user has, it's really a burden on the test developer to make sure that all the controls are working properly, that the accurate result is portrayed at the end of the day. So there's a lot of human factors, usability testing that is involved with the handheld disposable format, which inevitably takes a lot more time than what can be deployed with respect to the reagents themselves.
Great. We have a couple questions regarding the detection technology. The first one is, what is the viral genome copy limit for detection?
Yeah, so what we have published in our EUA that came out last month is about 20 copies per microliter for limit of detection. Since then, we've made a number of improvements that will be implemented in our high throughput testing platform. And the goal there is to, go below 500 copies per ml, so about, 0.5 copies per microliter. That really is in the same range as what we see with, gold standard RT-PCR assays.
Okay. And another related question is, are you targeting conserved regions of the virus for detection, considering that it's an RNA virus and mutations may occur? How do you deal with that?
Yeah, it's a really good question. So we have focused on conserved regions. We are fortunate as well that this particular virus doesn't mutate as quickly as some other RNA viruses. We also have strategies in the assay development that will enable us to, add additional guide RNAs as necessary to address relevant mutations that do arise in the future. But it is a critical piece of the development and design of these reagents to ensure that, you don't have any escape variants or mutants that arise. And so we are addressing that early on in the development.
Okay, I think we have time for one last question. This may be redundant, but what's the main selling point over Qpcr for a centralized lab? What do you have a leg up on?
Yeah, so there are a few areas. The first is faster turnaround time, right? So typically full process for RT-PCR can take somewhere between 4 to 6 hours from sample processing to results. Our solution is going to be much less than that. We're averaging about 2 hours from sample processing to result. It's going to be fully turnkey, meaning that the user simply puts in, or the clinical lab scientist puts in the sample, and the automation takes care of the remaining steps. The other piece as well, so faster turnaround time leads to higher throughput of testing, and we've also designed a solution to minimize the number of tips that are required per, let's say, run of a given sample lot. And so right now, many of us are talking to customers that have faced extreme shortages of consumables that are required for these testing platforms. And with the simpler workflow, we're designing into the workflow to really reduce the number of consumables that are required. So that's going to be very important as we continue to scale up the solution. And then the final thing is, again, this is an orthogonal reagent kit, which doesn't, pull on the supply chain of the existing Qpcr reagents. It's also going to be at a price point that's going to be the same or cheaper than what we find with the PCR-based methods.
Okay, thank you, Dr. Chen, for a very interesting seminar.
Thank you very much.